HOXD13 products join those of HOXA13 in the anterior region of the limb bud, while HOXA12, HOXA11, and HOXD1012 are expressed throughout the posterior two-thirds of the limb bud. HOXD13 products join those of HOXA13 in the anterior region of the limb bud, while HOXA12, HOXA11, and HOXD1012 are expressed throughout the posterior two-thirds of the limb bud. Hox genes in the evolution of the tetrapod limb. Macroevolution, the generation of morphological novelties and the evolution of new species in higher taxa, results from alterations of development. One of the most obvious macroevolutionary changes is that from the fish fin to the amphibian leg. As Ritchell in 1849 pointed out, there is considerable homology between the bones of the fish fin and the tetrapod limb, the pectoral and pelvic fins of the fish being homologous to the tetrapod forelimb and hind limb, respectively. While specific homologies were able to be made between the proximal elements of the fin and the limb, the homologies proposed between the autopod of the limb, the hand or foot at the distal end, and the rays of the fins, did not hold water. This was true even when one compared the tetrapod limb with the fins of the crossopterygian, lobe-finned fishes thought to have been closely related to the ancestors of the amphibians. While it seems to be homology for the proximal and central elements of the limb, the autopod seems to be something new what evolutionary biologists call a neomorphic structure. Recent studies have strongly suggested that the expression of the 5' genes of the Hox group may be crucial in the change from fin to limb. Tetrapods and fishes share the first two phases of the Hox expression pattern in their appendages. Thus, both groups form stylopods and zeugopods. However, the phase 3 pattern of Hox gene expression is unique to tetrapods and is not found in fishes. Moreover, this change in Hox gene expression is mediated by a single enhancer element that is not found in fishes. This phase 3 change represents an inversion of gene expression, placing the most 5' Hox gene products in the anterior of the limb bud. Instead of being restricted to the posterior of the limb bud, the expression of the 5' Hox gene sweeps across the distal mesenchyme, just beneath the ear. This band of expression is coincident with the digital arch from which the digits form. Thus, the Hox gene expression pattern is homologous between fish and tetrapod limbs in the proximal regions. The expression pattern in the late bud distal mesenchyme is new. Institute for Advanced Studies, Jodhpur 41. These studies are paleontological interpretations of Shubin and Alberch, who proposed that the path of digit formation was not, as previously believed, through the fourth digit making the fin rays homologous to the other digits, but through an arch of distal wrist condensations metapterygia that begins posteriorly and turns anteriorly across the distal mesenchyme. Thus, the border of 5' Hox D gene expression follows the metapterygial axis that Shubin and Alberch hypothesized as being the origin of digits. The foot and hand, then, appear to be new structures in evolution, and they appear to have been formed by the repositioning of Hox D gene expression during fin development. The use of the same enhancer to generate both fingers and toes also helped solve the problem of how these structures were evolved at the same time. Specification of the anterior-posterior limb axis, a zone of polarizing activity. The specification of the anterior-posterior axis of the limb is the earliest change from the pluripotent condition. In chicks, this axis is specified shortly before a limb bud is recognizable. Amundsen 938 showed that as early as the 16th somite stage, prospective wing mesoderm transplanted to the flank area develops into a limb with the anterior-posterior and dorsal-ventral polarities of the donor graft, not those of the host tissue. Although the division of the proximal distal structures is thought to depend on how many divisions a cell undergoes while in the progress zone, information instructing a cell as to its position on the anterior-posterior and dorsal-ventral axes must come from other sources. Several experiments suggest that the anterior-posterior axis is specified by a small block of mesodermal tissue near the posterior junction of the young limb bud and the body wall. This region of the mesoderm has been called the zone of polarizing activity, ZPA. Once taken from a young limb bud and transplanted into a position on the anterior side of another limb bud, the number of digits of the resulting wing is doubled. Moreover, the structures of the extra set of digits are mirror images of the normally produced structures. The polarity has been maintained, but the information is now coming from both an anterior and a posterior direction. Sonic Hedgehog defines the ZPA. The search for the molecules conferring this polarizing activity on the ZPA became one of the most intensive quests in developmental biology. In 1993, Riddle and colleagues showed by in situ hybridization that Sonic Hedgehog chish, a vertebrate homologue of the Drosophila hedgehog gene, was expressed specifically in that region of the limb bud known to be the ZPA. As the association between the ZPA and Sonic Hedgehog was more than just a correlation, RID 5060LE and coworkers, 1993, demonstrated that the secretion of Sonic Hedgehog protein is sufficient for ZPA activity. They transfected embryonic chick fibroblasts, which normally would never synthesize this protein, with a viral vector containing the shish gene. The gene became expressed and translated in these fibroblasts, which were then inserted under the anterior ectoderm of an early chick limb bud. Mirror image digit duplications like those induced by ZPA transplants were the result. More recently, beads containing Sonic Hedgehog protein were shown to cause the same duplications. Thus, Sonic Hedgehog appears to be the active agent of the ZPA. Specification of the posterior limb bud to express Sonic Hedgehog. Two new questions emerged from this discovery. First, how does Sonic Hedgehog become expressed only in the posterior region of the limb bud? And second, what does it do once it is expressed? We do not yet know what causes the activation of the Sonic Hedgehog gene specifically in the cells of the posterior limb bud and not in the cells located more anteriorly. The Sonic Hedgehog gene appears to be activated by an FGF protein coming from a newly formed apical ectodermal ridge. FGF8 is secreted from the air and is capable of activating Sonic Hedgehog. But why doesn't FGF8 activate all the mesenchyme cells beneath the air? The answer may reside in the differential competence of certain mesenchyme cells to respond to the FGF signal. Charite and colleagues 1994 have suggested that the HOXV8 protein may be critical in providing this restricted competence. They observed that the HOXV8 gene is usually expressed in the posterior half of the mouse forelimb bud. They then constructed transgenic mice in which the HOXV8 gene was placed under the control of a new promoter that would cause its expression throughout the forelimb buds. This resulted in the expression of Sonic Hedgehog in the anterior portion of the limb buds, the creation of a new ZPA, and mirror image forelimb duplications. This evidence suggests that the HOXV8 protein is involved in specifying the domain of Sonic Hedgehog expression and thus establishing the ZPA. Institute for Advanced Studies, Jodhpur 42. The action of Sonic Hedgehog. When Sonic Hedgehog was first shown to define the ZPA, it was thought to act as a morphogen. In other words, it was thought to diffuse from the ZPA where it was being synthesized and to form a concentration gradient from the posterior to the anterior of the limb bud. However, recent research has provided evidence that Sonic Hedgehog protein, or its active amino-terminal region, does not diffuse outside the ZPA. It is now thought that Sonic Hedgehog works by initiating and sustaining a cascade of other proteins, such as BMP2 and BMP7. A gradient of BMPs may emanate from the ZPA and specify the digits. However it works, Sonic Hedgehog directly or with help from the BMP cascade regulates the expression of the 5' Hox D genes. The transition from phase 1 to phase 2 Hox expression patterns is coincident with Sonic Hedgehog expression in the ZPA. Moreover, transplantation of either the ZPA or other Sonic Hedgehog secreting cells to the anterior margin of the limb bud at this stage leads to the formation of mirror image patterns of Hox-D gene expression and results in mirror image digit patterns. The generation of the dorsal ventral axis. The third axis of the limb distinguishes the dorsal limb knuckles, nails from the ventral limb pads, soles. In 1974, McAfee and co-workers demonstrated that the dorsal ventral polarity of the limb bud is determined by the ectoderm encasing it. The arm is rotated 180 degrees with respect to the limb bud mesenchyme. The dorsal-ventral axis is partially reversed. The distal element's digits are upside down. This suggested that the late specification of the dorsal-ventral axis of the limb is regulated by its ectodermal component. One molecule that appears to be particularly important in specifying the dorsal ventral polarity is WNT7A. The WNT7A gene is expressed in the dorsal but not the ventral ectoderm of the chicken mouse limb buds. In 1995, Parr and McMahon genetically deleted WNT7A from mouse embryos. The resulting embryos had sole pads on both surfaces of their paws, showing that WNT7A is needed for the dorsal patterning of the limb. Encodes a transcription factor that appears to be essential for specifying dorsal sulfates in the limb. If this factor is expressed in the ventral mesenchyme cells, they develop a dorsal phenotype. Mutants of LMX1 in humans and mice also show its importance for specifying dorsal limb fates. Now, this gene in mice produce a syndrome in which the dorsal limb phenotype is lacking, and loss of function mutations in humans produce a nail patella syndrome, a condition in which the dorsal sides of the limbs have been ventralized. Coordination among the three axes. The three axes of the tetrapod limb are all interrelated and coordinated. Some of the principal interactions among the mechanisms specifying the axes. Indeed, the molecules that define one of these axes are often used to maintain another axis. Hence, Sonic Hedgehog in the ZPA activates the expression of the FGF4 gene in the posterior region of the ear. FGF4 expression is important in recruiting mesenchyme cells into the progress zone, and it is also critical in maintaining the expression of Sonic Hedgehog in the ZPA. Therefore, the air and the ZPA mutually support each other through the positive loop of Sonic Hedgehog and FGF4. The WNT7A deficient mice mentioned above not only lacked dorsal limb structures, they also lacked posterior digits, suggesting that WNT7A is also needed for the anterior-posterior axis. Young and Niswander 1995 made a similar set of observations in chick embryos. These investigators removed the dorsal ectoderm from a developing limb and found that such an operation resulted in the loss of posterior skeletal elements from the limbs. The reason that these limbs lacked posterior digits was that Sonic Hedgehog expression was missing. Viral-induced expression of WNT7A was able to replace the dorsal ectoderm and restore Sonic Hedgehog expression and posterior phenotypes. These findings show that the synthesis of Sonic Hedgehog is stimulated by the combination of FGF4 and WNT7A proteins. Institute for Advanced Studies, Jodhpur 43. Cell death and the formation of digits in joints sculpting the autopod. Cell death plays a role in sculpting the limb. Indeed, it is essential if our joints are to form and if our fingers are to become separate. The death or lack of death of specific cells in the vertebrate limb is genetically programmed and has been selected for during evolution. One such case involves the webbing or non-webbing of feet. The difference between a chicken's foot and that of a duck is the presence or absence of cell death between the digits. Saunders and co-workers have shown that after a certain stage, chick cells between the digit cartilage are destined to die, and will do so even if transplanted to another region of the embryo or placed into culture. Before that time, however, transplantation to a duck limb will save them. Between the time when the cell's death is determined and when death actually takes place, levels of DNA, RNA, and protein synthesis in the cell decrease dramatically. In addition to the interdigital necrotic zone, there are three other regions that are sculpted by cell death. The ulnar radius are separated from each other by an interior necrotic zone, and two other regions, the anterior and posterior necrotic zones, further shape the end of the limb. Although these zones are said to be necrotic, this term is a holdover from the days when no distinction was made between necrotic cell death and apoptotic cell death. These cells die by apoptosis, and the death of the interdigital tissue is associated with the fragmentation of their DNA. The signal for apoptosis in the autopod is probably provided by the BMP proteins. BMP2, BMP4, and BMP7 are each expressed in the interdigital mesenchyme, and blocking BMP signaling by infecting progress zone cells with retroviruses carrying dominant negative BMP receptors prevents interdigital apoptosis. Since these BMPs are expressed throughout the progress zone mesenchyme, it is thought that cell death would be the default state unless there were active suppression of the BMPs. Suppression may come from a noggin protein, which is made in the developing digits and in the perichondrial cells surrounding them. If noggin is expressed throughout the limb bud, no apoptosis is seen. Forming the joints. The function originally ascribed to BMPs was the formation, not the prevention, of bone and cartilage tissue. In the developing limb, BMPs induce the mesenchymal cells either to undergo apoptosis or to become cartilage-producing chondrocytes depending on the stage of development. The same BMPs can induce death or differentiation, depending on the age of the target cell. This context dependency of signal action is a critical concept in developmental biology. It is also critical for the formation of joints. Lewis and colleagues 1997 have shown that during early limb bud stages before cartilage condensation, beads secreting BMP2 or BMP7 cause apoptosis. 2 days later, the same beads cause the limb bud cells to form cartilage. In the normally developing limb, BMPs use both these properties to form joints. BMP7 is made in the perichondrial cells surrounding the condensing chondrocytes and promotes cartilage formation. 2 other BMP proteins, BMP2 and GDF5, are expressed at the regions between the bones, where joints will form. Mouse mutations have suggested that the function of these proteins in joint formation is critical. Mutations of GDF5 produce brachydism, a condition characterized by a lack of limb joints. In mice homozygous for loss of functional alleles of noggin, no joints form, either. It appears that the BMP7 in these noggin defective embryos is able to recruit nearly all the surrounding mesenchyme into the digits. The roles of BMP2 and GDF5 are more controversial. They may either be destroying mesenchymal cells to form the joint or inducing them to rapidly differentiate and join one or the other cartilaginous nodule. In either way, a space is made between the nodules, and a joint can form. Limb development is an exciting meeting place for developmental biology, evolutionary biology, and medicine. We can expect to know the basis for numerous congenital diseases of limb formation, and perhaps we will understand how limbs are modified into flippers, wings, hands, and legs. Institute for Advanced Studies, Jodhpur 44. C4. Key points of developmental biology. A. Principles of development, DEVELOPMENTALANATOMY. 1. Organisms must function as they form their organs. They have to use one set of structures while constructing others. 2. The main question of development is, how does the egg becomes an adult? This question can be broken into the component problems of differentiation: how do cells become different from one another and from their precursors? Morphogenesis: how is ordered form is generated? Growth: how is size regulated? Reproduction: how does one generation create another generation? And evolution: how do changes in developmental processes create new anatomical structures? 3. Abigenesis happens. New organisms are created de novo each generation from the relatively disordered cytoplasm of the egg. 4. Preformation is not in the anatomical structures, but in the instructions to form them. The inheritance of the fertilized egg includes the genetic potentials of the organism. 5. The preformed nuclear instructions include the ability to respond to environmental stimuli in specific ways. 6. The ectoderm gives rise to the epidermis, nervous system, and pigment cells. 7. The mesoderm generates the kidneys, gonads, bones, heart, and blood cells. 8. The endoderm forms the lining of the digestive tube and the respiratory system. 9. Carl von Bayer's principles state that the general features of a large group of animals appear earlier in the embryo than do the specialized features of a smaller group. As each embryo of a given species develops, it diverges from the adult forms of other species. The early embryo of a higher animal species is not like the adult of a lower animal. 10. Living cells with dyes shows that some cells differentiate where they form, while others migrate from their original sites and differentiate in their new locations. Migratory cells include neural crest cells and the precursors of germ cells and blood cells. 11. Community of embryonic structure reveals community of descent, Charles Darwin. 12. Homologous structures in different species of those organs whose similarity is due to their sharing a common ancestral structure. Analogous structures of those organs whose similarity comes from their serving a similar function, but which are not derived from a common ancestral structure. 13. Anomalies can be caused by genetic factors, mutations, aneuploidies, translocations, or by environmental agents, certain chemicals, certain viruses, radiation. 14. Syndromes consist of sets of developmental abnormalities that run together. 15. Organs that are linked in developmental syndromes share either a common origin or a common mechanism of formation. 16. If growth is isometric, a twofold change in weight will cause a 1.26-fold expansion in length. 17. Allometric growth can create dramatic changes in the structure of organisms. 18. Complex may be self-generated by reaction diffusion events, wherein the activator of a local phenomenon stimulates the production of more of itself as well as the production of a more diffusible inhibitor. B. Life cycles and developmental patterns. 1. The life cycle can be considered a central unit in biology. The adult form need not be paramount. In a sense, the life cycle is the organism. 2. The basic life cycle consists of fertilization, cleavage, gastrulation, germ layer formation, organogenesis, metamorphosis, adulthood, and senescence. 3. Reproduction need not be sexual. Some organisms, such as volvox and dictyostelium, exhibit both asexual reproduction and sexual reproduction. 4. Cleavage divides the zygote into numerous cells called blastomeres. 5. In animal development, gastrulation rearranges the blastomeres and forms the 3 germ layers. 6. Organogenesis often involves interactions between germ layers to produce distinct organs. 7. Germ cells are the precursors of the gametes. Gametogenesis forms the sperm and the eggs. 8. There are 3 main ways to provide nutrition to the developing embryo. 1. Supply the embryo with yolk. 2. Form a larval feeding stage between the embryo and the adult. Or 3. Create a placenta between the mother and the embryo. 9. Life cycles must be adapted to the non-living environment and interwoven with other life cycles. 10. Don't regress your tail until you have formed your hind limbs. 11. There are several types of evidence. Correlation phenomenon A and phenomenon B does not imply that A causes B or that B causes a loss of function data. If A is experimentally removed, B does not occur, suggests that A causes B, but other explanations are possible. Gain of function data, if it happens where or when it does not usually occur, then B also happens in this new time or place, is most convincing. 12. Protozoans and deuterostomes represent 2 different sets of variations on development. Protozoans form the mouth first, while deuterostomes form the anus first. C. Experimental embryology. 1. There are norms of reaction that describe an embryo's inherited ability to develop a range of phenotypes. The environment can play a role in selecting which phenotype is expressed. Examples include temperature-dependent sex determination and seasonal phenotypic changes in caterpillars and butterflies. 2. Developing organisms are adapted to the ecological niches in which they develop. Examples include the ability of frog eggs exposed to sunlight to repair DNA damage. Institute for Advanced Studies, Jodhpur 45. 3. Before cells overtly differentiate into the many cell types of the body, they undergo a covert commitment to a certain fate. This commitment is first labile, the specification step, but later becomes irreversible, the determination step. 4. Autonomous specification. Removal of a blastomere from an embryo causes the absence in the embryo of those tissues formed by that blastomere. This mechanism of specification produces a mosaic pattern of development. Examples include early snail and tunicate embryos. 5. In autonomous specification, morphogenic determinants are apportioned to different blastomeres during cell cleavage. An example is the yellow crescent cytoplasm that is found in the muscle-forming cells of tunicate embryos. For by the other cells changing their fates, each cell can potentially give rise to more cell types than it normally does. This produces a regulative pattern of development wherein cell fates are determined relatively late. Examples include frog and mammalian embryos. 7. In conditional specification, the fate of a cell often depends upon its neighbors whom it meets. 8. In conditional specification, groups of cells can have their fates determined according to a concentration gradient of morphogen. The cells specified by such a morphogen can constitute a field. 9. In syncretional specification, the fates of cells can be determined by gradients of morphogens within the egg cytoplasm. 10. Different cell types can sort themselves into regions by means of cell surface molecules such as cadherins. These molecules can be critical in patterning cells into tissues and organs. D. Genes in development. 1. Development connects genotype and phenotype. 2. Nuclear genes are not lost or mutated during development. The genome of every cell is equivalent. 3. The exceptions to the rule of genomic equivalence are the lymphocytes. During differentiation, these cells rearrange their DNA to create new immunoglobulin and antigen receptor genes. 4. Genetic equivalence is implied by metaplasia, in which one differentiated cell type becomes another differentiated cell type. An example is the transdifferentiation of a salamander dorsal iris into a lens when the lens is removed. 5. Nuclei from differentiated cells to direct the development of complete adult organisms has recently confirmed the principle of genomic equivalence. 6. The cloning of beings, as well as regenerating damaged organs or enhancing physical abilities, may soon be possible through cloning technology and the use of embryonic stem cells. 7. Only a small percentage of the genome is expressed in any particular cell. 8. Chromosomes, in which the DNA has replicated without separating, as in larval Drosophila salivary glands, show regions where DNA is being transcribed. Different cell types show different regions of DNA being transcribed. 9. Northern blots, in situ hybridization, and the polymerase chain reaction can show which cells are transcribing particular genes. 10. Functions of a gene often can be ascertained by antisense mRNA, transgenic expression, or, in the case of mammals, gene knockouts. 11. Knowledge of gene activity in humans can be obtained by candidate gene mapping or positional cloning. E. Developmental genetics. 1. Differential gene expression from genetically identical nuclei creates different cell types. Differential gene expression can occur at the levels of gene transcription, nuclear RNA processing, mRNA translation, and protein modification. 2. Genes are usually repressed. Activation of a gene often means inhibiting its repressor. Leads to thinking in double and triple negatives. Activation is often the inhibition of the inhibitor. Repression is the inhibition of the inhibitor of the inhibitor. 3. Eukaryotic genes contain promoter sequences to which RNA polymerase can bind to initiate transcription. The eukaryotic RNA polymerases are bound by a series of proteins called basal transcription factors. 4. Eukaryotic genes expressed in specific cell types contain enhancer sequences that regulate their transcription in time and space. 5. Specific transcription factors can recognize specific sequences of DNA in the promoter and enhancer regions. They activate or repress transcription from the genes to which they have bound. 6. Enhancers work in a combinatorial fashion. The binding of several transcription factors can act to promote or inhibit transcription from a certain promoter. In some cases, transcription is activated only if both factor A and factor B are present, while in other cases, transcription is activated if either factor or factor B is present. 7. A gene encoding a transcription factor can keep itself activated if the transcription factor it encodes also activates its own promoter. A transcription factor gene can have one set of enhancer sites to initiate its activation and a second set of enhancer sites that bind the encoded transcription factor to maintain its activation. 8. Often, the same transcription factors that are used during the differentiation of a particular cell type are also used to activate the genes for that cell type specific products. For instance, PAX6 is needed both for the differentiation of the lens and for the transcription of the lens crystalline genes, and MITF is needed for pigment cell differentiation and for the transcription of the genes whose products catalyze the synthesis of melanin. 9. Enhancers can act as silencers to suppress the transcription of a gene in inappropriate cell types. 10. Locus control regions may function by making relatively large portions of a chromosome accessible to transcription factors. 11. Transcription factors act in different ways to regulate RNA synthesis. Some transcription factors stabilize RNA polymerase binding to the DNA, some disrupt nucleosomes, and some increase the efficiency of transcription. 12. Transcription correlates with a lack of methylation on the promoter and enhancer regions of genes. Methylation differences can account for examples of genomic imprinting, wherein a gene transmitted through the sperm. Institute for Advanced Studies, JADPR 46. Is expressed differently from the same gene transmitted through the egg. 13. Dosage compensation enables the X chromosome-derived products of males, which have one X chromosome per cell in fruit flies and mammals, to equal the X chromosome-derived products of females, which have two X chromosomes per cell. This compensation is accomplished at the level of transcription, either by accelerating transcription from the lone X chromosome in males Drosophila, or by inactivating a large portion of one of the two X chromosomes in females mammals. 14. X chromosome inactivation in placental mammals is generally random and involves the activation of the cis gene on the chromosome that will be inactivated. 15. Differential RNA selection can allow certain transcripts to enter the cytoplasm while preventing other transcripts from leaving the nucleus. 16. Differential RNA splicing can create a family of related proteins by causing different regions of the nRNA to be read as exons and introns. What is an exon in one set of circumstances may be an intron in another. 17. Messages are translated only at certain times. The oocyte, in particular, uses translational regulation to set aside certain messages that it transcribes during egg development but uses only after the egg is fertilized. This activation is often accomplished either by the removal of inhibitory proteins or by the polyadenylation of the message. 18. Many messenger RNAs are localized to particular regions of the oocyte or other cells. This localization appears to be regulated by the 3' untranslated region of the mRNA. F. Cell-cell communication. 1. Inductive tissue interaction involves inducer and responding tissues. 2. The ability to respond to inductive signals depends upon the competence of the responding cells. 3. Reciprocal induction occurs when the two interacting tissues are both inducers and are competent to respond to each other's signals. 4. Cascades of inductive events are responsible for organ formation. 5. Regionally specific inductions can generate different structures from the same tissue. 6. The ability to respond to inducers is determined by the genetic state of the responding tissue. 7. Ductrine interactions are inductions that occur between the cell membranes of adjacent cells or between a cell membrane and an extracellular matrix secreted by another cell. 8. Paracrine interactions occur when a cell or tissue secretes proteins that induce changes in neighboring cells. 9. Paracrine factors are inducing proteins that bind to cell membrane receptors in competent responding cells. 10. Competent cells respond to paracrine factors through signal transduction pathways. Competence is the ability to bind and to respond to the inducers, and it is often the result of a prior induction. 11. Signal transduction pathways begin with the paracrine or juxtacrine factor causing a conformational change in its cell membrane receptor. The new shape results in enzymatic activity in the cytoplasmic domains of the receptor protein. This allows the receptor to phosphorylate other cytoplasmic proteins, thereby activating a dormant kinase activity. Actually, a transcription factor, or set of factors, is activated that activates or represses specific gene activity. 12. Pleiotropy is the phenomenon of many phenotypic changes being caused by one mutation. Mosaic pleiotropy results when the mutant gene is used in different parts of the body and each part is separately altered. Relational pleiotropy occurs when a particular defect caused by the mutant gene affects other parts of the body that do not express the gene. 13. Genetic heterogeneity results when multiple genes are needed to create a particular phenotype. When mutant genes for a paracrine factor cause syndromes similar to those generated by mutant genes for the factor's receptor. 14. Phenotypic heterogeneity results when the same mutation produces different phenotypic effects in different individuals. It is caused by the interactions between gene products. 15. Dominant mutation, in which only one mutant gene of the diploid pair is necessary to produce an abnormal phenotype, can be caused by haploinsufficiency, gain-of-function mutations, or dominant negative alleles. 16. Programmed cell death is one possible response to inductive stimuli. Apoptosis is a critical part of life. 17. There is crosstalk between signal transduction pathways, which allows the cell to respond to multiple inputs simultaneously. G. Fertilization. 1. Fertilization accomplishes two separate activities: sex, the combining of genes derived from two parents, and reproduction, the creation of a new organism. 2. The and include 1. Contact and recognition between sperm and egg, 2. Regulation of sperm entry into the egg, 3. Fusion of genetic material from the two gametes, and 4. Activation of egg metabolism to start development. 3. The sperm head consists of a haploid nucleus and an acrosome. Acrosome is derived from the Golgi apparatus and contains enzymes needed to digest extracellular coats surrounding the egg. The neck of the sperm contains the mitochondria and the centriole that generates the microtubules of the flagellum. Energy for flagellar motion comes from mitochondrial ATP and a dynein ATPase in the flagellum. 4. The egg contains a haploid nucleus and an enlarged cytoplasm storing ribosomes, mRNAs, and nutritive proteins. Other mRNAs and proteins, used as morphogenic factors, are also stored in the egg. Cortical granules lie beneath the egg's plasma membrane. Many eggs also contain protective agents needed for survival in their particular environment. 5. Surrounding the egg plasma membrane is an extracellular layer often used in sperm recognition. In most animals, this extracellular layer is the vitelline envelope. In mammals, it is a much thicker zona pellucida. 6. In many species, eggs secrete diffusible molecules that attract and activate the sperm. 7. In sea urchins, the acrosome reaction is initiated by compounds in the egg jelly. The acrosomal vesicle undergoes exocytosis to release its enzyme. Globular actin polymerizes to extend the acrosomal process. Bindin on the acrosomal process is recognized by a protein complex on the sea urchin egg surface. Institute for Advanced Studies, Jodhpur 47. 8. In mammals, sperm must be capacitated in the female reproductive tract before they are capable of fertilizing the egg. 9. Mammalian sperm bind to the zona pellucida before undergoing the acrosome reaction. In them, this binding is mediated by ZP3, zona protein 3, and one or many sperm proteins that recognize it. The mammalian acrosome reaction is initiated on the zona pellucida, and the acrosomal enzymes are concentrated there. 10. In between sperm and egg is mediated by protein molecules whose hydrophobic groups can merge the sperm and egg plasma membranes. In sea urchins, bindin may mediate gamete fusion. In mammals, fertilin proteins in the sperm bind to integrins in the egg and allow the membranes to fuse. Polyspermy results when two sperm fertilize the egg. It is usually lethal, since it results in 3 sets of chromosomes divided among 4 cells. 12. There are often 2 blocks to polyspermy. The fast block is electrical and is mediated by sodium ions. The egg membrane resting potential rises, and sperm can no longer fuse with the egg. The slow block is physical and is mediated by calcium ions. A wave of calcium ions propagates from the point of sperm entry, causing the cortical granules to fuse with the egg cell membrane. The released contents of the granules cause the vitelline membrane to rise and to harden into the fertilization envelope. 13. In mammals, blocks to polyspermy include the modification of the zona proteins by the contents of the cortical granule. Sperm can no longer bind to the zona. 14. 1,4,5-triphosphate IP3 is believed to be responsible for releasing calcium ions from storage in the endoplasmic reticulum. DAG-diacylglycerol is thought to initiate the rise in egg pH. The free calcium ions, supported by the alkalization of the egg, activate egg metabolism, protein synthesis, and DNA synthesis. 15. The male pronucleus and the female pronucleus migrate toward each other, replicating DNA as they move. 16. In sea urchins, the two pronuclei merge and a diploid zygote nucleus is formed. In mammals, the pronuclei disintegrate as they approach each other, and their chromosomes gather around a common metaphase plate. 17. Some genes are transmitted differently depending on whether they are from the egg or the sperm. Methylation differences determine if these genes are to be expressed in the early embryo. 18. Microtubular changes cause cytoplasmic movements. These rearrangements of cytoplasm can be critical in specifying which portions of the egg are going to develop into which organs. H. Early invertebrate development. 1. During cleavage, most cells do not grow. Rather, the volume of the oocyte is cleaved into numerous cells. The major exceptions to this rule are mammals. 2. The plasmid cell cycle is governed by the synthesis and degradation of cyclin. Cyclin synthesis promotes the formation of MPF, and MPF promotes mitosis. Degradation of cyclin brings the cell back to the S phase. G phases are added at the mid-blastula transition. 3. Blast. Vocabulary. A blastomere is a cell derived from cleavage in an early embryo. A blastula is an embryonic structure composed of blastomes. The cavity in the blastula is the blastocele. If the blastula lacks a blastocele, it is a stereoblastula. A mammalian blastula is called a blastocyst in chapter 11, and the invagination where gastrulation begins is the blastopore. 4. Movements of gastrulation include invagination, involution, ingression, delamination, and epiboly. 5. 3 accommodations of the body. The anterior-posterior axis, head to tail or mouth to anus, the dorsal-ventral axis back to belly, and the right-left axis between the two lateral sides of the body. 6. In all 4 invertebrates described here, cleavage is holoblastic. In the sea urchin, cleavage is radial, in the snail, spiral, in the tunicate, bilateral, and in the nematode, rotational. 7. In the tunicate, snail, and nematode, gastrulation occurs when there are relatively few cells, and the blastopore becomes the mouth. This is the protostome mode of gastrulation. 8. Body axes in these species are established in different ways. In some, such as the sea urchin and tunicate, the axes are established at fertilization through determinants in the egg cytoplasm. In other species, such as the nematode and snail, the axes are established by cell interactions later in development. 9. In the sea urchin, gastrulation occurs only after thousands of cells have formed, and the blastopore becomes the anus. This is the deuterostome mode of gastrulation, and is characteristic only of echinoderms and chordates. 10. In sea urchins, cell fates are determined by signaling. The micromers constitute a major signaling center. Beta-catenin is important for the inducing capacity of the micromers. 11. Differential cell adhesion is important in regulating sea urchin gastrulation. The micromers delaminate first from the vegetal plate. They form the primary mesenchyme which becomes the skeletal rods of the Pluteus larva. The vegetal plate invaginates to form the endodermal archenteron, with a tip of secondary mesenchyme cells. The archenteron elongates by convergent extension and is guided to the future mouth region by the secondary mesenchyme. 12. Snails exhibit spiral cleavage and form stereoblastulae, having no blastoceles. The direction of the spiral cleavage is regulated by a factor encoded by the mother and placed into the oocyte. Spiral cleavage can be modified by evolution, and adaptations of spiral cleavage have allowed some mollusks to survive in otherwise harsh conditions. 13. The polar lobe of certain mollusks contains the determinants for mesoderm and endoderm. These will enter the D blastomere. 14. The tunicate fate map is identical on its right and left sides. The yellow cytoplasm contains muscle-forming determinants. These act autonomously. The nervous system of tunicates is formed conditionally, by interactions between blastomeres. 15. The Sloanorhabditis elegans was chosen as a model organism because it has a small number of cells, a small genome, is easily bred and maintained, has a short lifespan, can be genetically manipulated, and has a cuticle through which one can see cell movements. 16. In the organs of the C. elegans zygote, one daughter cell becomes a founder cell, producing differentiated descendants, and the other becomes a stem cell, producing other founder cells in the germline. Institute for Advanced Studies, Jodhpur 48. 17. Blastomere identity in C. elegans is regulated by both autonomous and conditional specification. I. Drosophila development and axis specification. 1. Drosophila cleavage is crucial. The nuclei divide 13 times before forming cells. Before cell formation, the nuclei reside in a syncatial blastome. Each nucleus is surrounded by an actin-filled cytoplasm. 2. When the cells form, the Drosophila embryo undergoes a midblastula transition, wherein the cleavages become asynchronous and new mRNA is made. The amount of chromatin determines the timing of this transition. 3. Gastrulation begins with the invagination of the most ventral region, the presumptive mesoderm. This causes the formation of a ventral furrow. The germ band expands such that the future posterior segments curl just behind the presumptive head. 4. Maternal effect genes are responsible for the initiation of anterior-posterior polarity. Bicoid mRNA is sequestered by its 3' UTR in the future anterior by the cytoskeleton. Nanos mRNA is sequestered by its 3' UTR in the future posterior pole. Hunchback and caudal messages are seen throughout the embryo. 5. At fertilization, bicoid and nanos messages are translated. A gradient of bicoid protein activates more hunchback transcription in the anterior. Moreover, bicoid inhibits the translation of caudal mRNA. A gradient of nanos in the posterior inhibits the translation of hunchback mRNA. Caudal protein is made in the posterior. 6. Bicoid and hunchback proteins activate the genes responsible for the anterior portion of the flight. Caudal activates genes responsible for posterior development. 7. The unsegmented anterior and posterior are regulated by the activation of the torso protein at the anterior and posterior poles of the egg. 8. The gap genes respond to concentrations of the maternal effect gene proteins. Their protein products interact with each other such that each gap gene protein defines specific regions of the embryo. 9. The gap gene proteins activate and repress the pair rule genes. The pair rule genes have modular promoters such that they become activated in the 7 stripes. Their boundaries are defined by the gap genes. These genes form 7 bands of transcription along the anterior-posterior axis, each one comprising 2 parasegments. 10. The pair rule gene products activate engrailed and wingless expression in adjacent cells. The engrailed expressing cells form the anterior boundary of each parasegment. These cells form a signaling center that organizes the cuticle formation and segmental structure of the embryo. 11. The homeotic selector genes are found in 2 complexes on chromosome 3 of Drosophila. Together these are called home C, the homeotic gene complex. The genes are arranged in the same order as their transcriptional expression. These genes specify each segment, and mutations in these genes are capable of transforming one segment into another. 12. The expression of each homeotic selector gene is regulated by the gap and pair rule genes. Their expression is refined and maintained by interactions whereby the protein products interact with genes, preventing the transcription of neighboring home C genes. 13. In ultravithorax mutations, the third thoracic segment becomes specific as the second thoracic segment. This converts the halteres into wings. When antennapedia is expressed in the head as well as in the thorax, it represses antenna formation, allowing legs to form where the antenna should be. 14. The targets of the home C proteins are the relicitator genes. These include distal-less and wingless genes in the thoracic segments. 15. Dorsal ventral polarity is regulated by the entry of the dorsal protein into the nucleus. Dorsal polarity is initiated by the nucleus being positioned in the dorsal anterior of the oocyte and transcribing the Gurkin message. This message is transported to the region above the nucleus and adjacent to the follicle cells. 16. The Gurkin mRNA is translated into the Gurkin protein, which is secreted from the oocyte and binds to its receptor, torpedo, on the follicle cells. This dorsalizes the follicle cells, preventing them from synthesizing pipe. 17. The pipe protein in the ventral follicle cells modifies an as yet unknown factor that modifies the noodle protein. This allows the noodle protein to activate a cascade of proteolysis in the space between the ventral follicle cells and the ventral cells of the embryo. 18. As a result of the cascade, the Spatzel protein is activated and binds to the toll protein on the ventral embryonic cells. 19. Activated toll protein activates PEL and tube to phosphorylate the cactus protein, which has been bound to the dorsal protein. Phosphorylated cactus protein is degraded, allowing dorsal protein to enter the nucleus. 20. When nucleus, dorsal protein activates the genes responsible for the ventral sulfates and represses those genes whose proteins would specify dorsal sulfates. Since a gradient of dorsal protein enters the various nuclei, those at the most ventral surface become mesoderm, those more lateral become neurogenic ectoderm. 21. Organs form at the intersection of dorsal ventral and anterior posterior regions of gene expression. J. Early development and axis FORMATIONIN amphibians. 1. Amphibian cleavage is holoblastic, but unequal due to the presence of yolk in the vegetal hemisphere. 2. Amphigastrulation begins with the invagination of the bottle cells, followed by the coordinated involution of the mesoderm and the epiboly of the ectoderm. Vegetal rotation plays a significant role in directing the involution. 3. Driving forces for ectodermal epiboly and the convergent extension of the mesoderm are the intercalation events in which several tissue layers merge. Fibronectin plays a critical role in enabling the mesodermal cells to migrate into the embryo. 4. The dorsal lip of the blastopore forms the organizer tissue of the amphibian gastrula. This tissue dorsalizes the ectoderm, transforming it into neural tissue, and it transforms ventral mesoderm into lateral mesoderm. 5. The organizer consists of pharyngeal endoderm, head mesoderm, notochord, and dorsal blastopore lip. The organizer functions by secreting proteins noggin, chordin, and follistatin that block the BMP signal that. Institute for Advanced Studies, Jodhpur 49. Would otherwise ventralize the mesoderm and activate the epidermal genes in the ectoderm. 6. In the head, an addition set of proteins Cerberus, FRZB, tick-off block the WNT signal from the ventral and lateral mesoderm. 7. The organizer is itself induced by the Nucleus center, located in the dorsalmost vegetal cells. This center is formed by cortical rotation during fertilization, which translocates the disheveled protein to the dorsal side of the egg. 8. The disheveled protein stabilizes beta-catenin in the dorsal cells of the embryo. Thus, the Nucleus center is formed by the accumulation of beta-catenin, which can complex with TCF3 to form a transcription factor complex that can activate the transcription of the CMY gene. 9. The CMY product and a TGF beta signal, perhaps from VG1, can activate the goosecoid gene in the organizer. The goosecoid gene can activate other genes that cause the organizer to function. 10. Other posteriorizing signals WNT3A, retinoic acid, EFGF can influence the anterior-posterior specification of the neural tube. 11. The left-right axis appears to be initiated at fertilization through the VG1 protein. In a still unknown fashion, this protein activates a nodal protein solely on the left side of the body. As in other vertebrates, the nodal protein activates expression of PIDX2, which is critical in distinguishing left-sidedness from right-sidedness. In the heart and gut tubes. K. The early development of vertebrates. 1. Reptiles and birds undergo discoidal meroblastic cleavage, wherein the early cell divisions do not cut through the yolk of the egg. These cells form a blastoderm. 2. In fishes, the deep cells form between the yolk synchytial layer and the enveloping layer. These cells migrate over the top of the yolk, forming the hypoblast and epiblast layers. On the future dorsal side, these layers intercalate to form the embryonic shield, a structure homologous to the amphibian organizer. Transplantation of the embryonic shield into the ventral side of another embryo will cause the formation of a second embryonic axis. 3. There are to be two signaling centers supplying anterior-posterior information in fishes, one located at the border between the neural and surface ectoderm,